Welcome to MU's World Biology Lecture 1. Today's topic is endosymbiosis, which is related with evolutionary biology.
Approximately 1 billion years ago, other cells absorbed cyanobacteria that eventually became chloroplasts, organelles that produce energy from sunlight. Some 100 million years ago, UCYNA, a nitrogen-fixing bacteria, became an endosymbiont of marine algae Brachydisphera bigeloii, eventually evolving into a nitroplast. An endosymbiont is one organism that lives inside of another one. All eukaryotic cells, like your own, are creatures that are made up of the parts of other creatures. Mitochondria, the important energy generators of our cells, evolved from free-living cells.
Mitochondria are best known for their role in the generation of ATP by aerobic respiration. Yet, research in the past half-century has shown that they perform a much larger suite of functions, and that these functions can vary substantially among diverse eukaryotic lineages. Despite this diversity, all mitochondria derive from a common ancestral organelle that originated from the integration of an endosymbiotic alpha proteobacterium into a host cell related to Ascaridia. The transition from endosymbiotic bacterium to permanent organelle entailed a massive number of evolutionary changes, including the origins of hundreds of new genes, and a protein import system, insertion of membrane transporters, integration of metabolism and reproduction, genome reduction, endosymbiotic gene transfer, lateral gene transfer, and the retargeting of proteins. These changes occurred incrementally as the endosymbiont and the host became integrated. Although many insights into this transition have been gained, controversy persists regarding the nature of the original endosymbiont, its initial interactions with the host, and the timing of its integration relative to the origin of other features of eukaryote cells. Since the establishment of the organelle, proteins have been gained, lost, transferred, and retargeted as mitochondria have specialized into the spectrum of functional types seen across the eukaryotic tree of life. The endosymbiotic theory of the origin of chloroplasts and mitochondria explains that chloroplasts originated from an ancient endosymbiotic cyanobacterial cell, and that mitochondria from an ancient endosymbiotic alpha proteobacterial cell. The theory was first proposed in a clear form by Merezhkovsky or chloroplast origin, but remained a mysterious, unsupported hypothesis during the first half of the 20th century. The similarity of oxygenic photosynthesis involving chlorophylls, the presence of internal membranes later called thylakoid membranes, the presence of peptidoglycan in some cyanelles, and later the presence of DNA in chloroplasts constituted the initial basis of the endosymbiotic origin of chloroplasts. The results of phylogenetic analysis of ribosomal RNA, ribosomal proteins, and various other proteins encoded by the chloroplast genome clearly showed the close relationship between chloroplasts and cyanobacteria, and, after critical examination, were taken as good evidence for the endosymbiotic origin of chloroplasts. The chromosome is the genetic material of the bacterium. Genes located along the DNA are transcribed into RNA molecules, primarily messenger RNA, mRNA, transfer RNA, tRNA, and ribosomal RNA, RNA. Messenger RNA is then translated into protein at the ribosomes. The ribosome is 70S, and other side, the mitochondrial and chloroplast ribosomes are also 70S. Now we will taking about the reproduction process of bacteria, chloroplast, and mitochondria. Binary fission is the process of one cell simply dividing into two. It is most commonly used in archaea and bacteria, which are both prokaryotic organisms, but it is also used in some eukaryotic protists. Binary fission is a type of asexual reproduction, where the offspring are genetic clones of the parents. Mitochondrial DNA has a simple circular structure, which is structurally similar to bacteria DNA and is more or less the same size. Mitochondrial ribosome is 70S. Mitochondrial ribosomes, enzymes, and transport systems are all similar to those of bacteria. Moreover, mitochondria are approximately the same size. Mitochondria divide by binary fission. Since mitochondria were created by bacterial endosymbiosis, they divide using a method similar to prokaryotic binary fission. Cycles of fission and fusion occur continuously in mitochondria. Mitochondrial fission is a multi-step process allowing the division of one mitochondrion in two daughter mitochondria. It is regulated by the recruitment of the GTPase dynamin-related protein 1, DRP1, by adapters at actin and endoplasmic reticulum-mediated mitochondrial constriction sites. Same in chloroplast. Subscribe the channel for more interesting topics.